welcome you all to the class of drug discovery and drug designing. Dear students, today we are going to learn about protein structure. In the last class we have discussed about the target identification, target validation and drug and lead identification. There is great role of the target structure prediction, target protein of the pathogenic pathogens, bacteria or virus. So protein structure play an important role. It is very important to know about the protein structure and function before targeting them, because proteins are the target from the pathogenic proteins, pathogenic bacteria and viruses. So it is really very important to know about the protein because these are the targets. So before going into the details of structure, this is the introduction of the protein. Proteins are the major components of all cellular systems. Proteins consist of one or more linear polymers, that is called polypeptide, means it is made up of the peptide chain. So different peptide connected with each other, then that makes the complete polypeptide chain. Proteins are linear and never branched. Different amino acids are linked together via peptide bonds. Okay. So each individual amino acid, that is called residue. So this is the example of oxytocin, is the example of the smallest protein. And largest protein is about 25,000 residue long. Okay. The name of that protein is titin. Proteins are generally between 100 to 1000 residue in length, means this is the length of the general proteins. In the absence of stabilizing forces, a minimum of 40 residues is needed to adopt a stable 3D structure in water. This means we can say that this will make the domain structure, because domain structure is only the stable structure in the proteins, tertiary structure. So minimum 40 amino acids are required. So we will be talking about the domain structure also. In your syllabus you have the domain and motif topic. Okay. So protein sequence can be determined by systematically removing amino acid. So there is a method that is called admin degradation. Okay. Protein sequence is important because this is encoded by the genes, and genetic code play an important role in that. Relationship between the gene and the amino acid is known as genetic code. So each amino acid has same fundamental structure, differing only in side chain designated the R groups, the carbon atom to which the amino acid carboxyl group and side chain are attached in the alpha carbon. So this is about the structure of the proteins. So in the linear structure, amino acids are connected with each other by the peptide bond. And there are some presence of R group also, that makes the different interaction with the other side chains of the amino acids. So these R groups are attached to the alpha carbon atom. These are all the amino acids that are found within the protein: aspartic acid, glutamic acid. These are negatively charged amino acids. These are also known as acidic amino acids: arginine, lysine, histidine. These are positively charged amino acids, and that are arginine, lysine and histidine. These are positively charged amino acids. So these amino acids have basic nature. So there is another category. Okay, first we will talk about the non-polar amino acid. On the right hand side you can give the list of the amino acids that are non-polar amino acid. In that category also there is the two category that are one category has the aromatic moiety and another category contains the aliphatic chain. So here you can see the tryptophan, phenylalanine, proline. Okay. So these are the amino acid okay, which contain the aromatic moiety, aromatic ring, you can say ring structure. And there are the other amino acid which contain the aliphatic chain. So these all are the non-polar amino acid. Alanine, glycine, valine, leucine, isoleucine. So these all amino acid contain the aliphatic chain. Okay, and these are called non-polar amino acid. So other than these amino acids which we have talked about, all are known as like polar amino acids. So on the left hand side we can see these all are the polar amino acid. In this also we were talking about the acidic amino acid and basic amino acid. Okay, so the amino acids that are shown in the sky blue color, these are known as acidic amino acid. Arginine, lysine and histidine, these are known as basic amino acid. And other than these, among the polar amino acids, so all are polar amino acids. So these are wholly polar, means which has the hydrophilic nature. Okay, so on the basis of this table we can make the 4 groups. Means the 4 groups are visible. You can see in the 4 colors, these are 4 groups. Within the polar also we have the 3 groups: acidic amino acid, basic amino acids and okay other non-polar amino acids. Okay, so we need to learn about the structure of the protein. We need to learn about the structure of all 20 amino acids. Okay, 3-letter abbreviations generally we denote in the docking or when we dock the drugs with drug structure with the protein. So we need that. Okay, and 1-letter code of the, so these things need to be remembered. Peptide bond formation, so there is a peptide bond formation between 2 amino acids. So here you can see these are the 2 amino acids, glycine and alanine, and there is a peptide bond formation between these 2 amino acids. Okay, so one amino acid contain NH2 group, another amino acid contain carboxylic group. Okay, that is negatively charged. So after peptide bond formation, water is released. So there is a peptide bond formation. So in that you can see glycyl alanine is formed here. So this is a peptide bond formation. This is the condensation reaction. So water is produced in this reaction. Here you can see these are the structures of different amino acids like methionine, okay, leucine. So side chains are shown in the colors. Like here you can see in the leucine we have CH2, CH, CH3, CH3. We have 2 CH3 groups, methyl groups. One is CH2 group and another one is CH. Okay, so in the different amino acids you can see there are the presence of R side chain that are shown in the color. Okay, in the structure of tyrosine you can see there is the aromatic ring structure. So here in the structure of aspartic acid also you can see there is the aliphatic chain. So here clearly you can see there is a difference between the tyrosine structure, okay, and aspartic acid structure. So polypeptide backbone contains the side chain, okay, amino terminus, carboxyl group terminus, and there is a between the 2 amino acids there is the formation of peptide bond. Here you can see. Okay, side chains are shown in the different color. So there is the formation of non-covalent bond between the chains, p chains. These are important in their overall conformation and function. So here you can see there is the formation of ion. There are different types of bonds that are found in the protein structure. Van der Waals interaction, hydrogen bonds, these are important. Ionic bond, these all bonds are important. So first of all, before going on to the complicated bond like ionic bond or other bonds, interactions are important. Van der Waals interactions and electrostatic interactions are important. Okay, there are other bonds also, like disulfide bond, that are also important in the tertiary structure. But in the primary structure and secondary structure, ionic bond, hydrogen bond, van der Waals interactions are important. For hydrogen bond to occur, there should be the presence of van der Waals forces and electrostatic interaction. Collectively, they play an important role for hydrogen bond formation. Like electrostatic interaction is important. Okay. So here, so what is important for the hydrogen bonds? If there is a charge difference, atoms attract with each other. That is the electrostatic interaction, because all the atoms have the charge differences like +2, +1, -2, -1. So if the atoms have the same charge like negative, they can also be attracted towards each other due to the presence of electrostatic interaction. Why this occurs? Due to the charge difference. Positive ions can also be attracted to each other. Why? Because of charge difference. So that is known as electrostatic interaction. What is van der Waals interaction? This is also important interaction. Nucleus of one atom is attracted towards the electron of another atom. What is found in the nucleus? Proton. Proton has the positive charge and it is attracted towards electrons of another atom. So the side groups of the linear unfolded polypeptides are intermingled only when correctly folded. So here you can see this is a tertiary structure of protein. How it is made up? So on the right hand side you can see the tertiary structure of protein, how it is folded, and on the left hand side you can see the unfolded polypeptide. So here in the unfolded polypeptide you can see polar chains and non-polar chains that are found in the core region of protein. In the right hand side, on the right hand side you can see hydrophobic core region contain non-polar side chain that is inside the protein. It is found in the core structure. We do not face the water or aqueous environment. So within the aqueous environment, all the hydrophobic chain face towards the core region. Hydrogen bonds can be formed to the polar side chains on the outside of the molecule. So hydrogen bonds play an important role within the aqueous environment. So in the body we have the aqueous environment, so that's why it is responsible for the formation of folded structure. Energy should be negative. The Gibbs free energy play an important role in the stabilization of this structure. This reaction is spontaneous because in our cells we have this kind of environment. Free energy is important. Minimum energy. Structure is formed on the minimum energy and maximum entropy. So there is a more negative enthalpy, means delta H should be more negative for the delta G to be negative. Then this change become spontaneous. What happened in the unfolded structure? If you want this structure to be fold, so we need the energy, like means there should be proper hydrogen bonds. Then the structure will get fold. Okay, so energy difference between these two structure is the amount of energy that is required to make the folded structure unfolded. Okay, so that will give you the idea about delta G free energy or enthalpy. Location of hydrogen bonds are not restricted to these between the sites. So as I have already told you, there are the other bonds also, van der Waals interaction and electrostatic interaction. These all interactions play an important role in the formation of hydrogen bonds. And these are not only restricted to the side chains. So this is the role of the van der Waals interaction. Here you can see, so there are the presence of van der Waals interaction. So here in this structure you can see how the van der Waals interaction play an important role, because this makes the atoms to come closer so that they can make the hydrogen bonds. So this is the role of the van der Waals interactions. In electrostatic interaction, atoms can come close to each other. So at the close distance they will make the hydrogen bonds. If they cross the atom cross the van der Waals radius, then the atoms feel repulsion. That is responsible for the steric hindrance, repulsion. Okay, and that some atoms are becomes out of conformation. So here you can see the folded structure and folded structure. So energy is important. Energy should be minimum for the folded structure. So this occur in the nature. This is a wonder of the nature, how it happens. So in the generally in our body, in the biological system, all the reactions are coupled reaction, like endergonic reaction and exergonic reaction. In one reaction, the energy is produced in the form of ATP. In the another reaction, energy is utilized. ATP is hydrolyzed. So the biological reactions are coupled reactions. So energy is required so that the reaction can become spontaneous, or reaction can be proceeded in the biological system. So any change, if there is a positive, the delta G energy is positive, gas free energy is, if that is a positive for any reaction, that reaction will not be spontaneous. So it needs the ATP. Okay. So but here the bonds play an important role. Location of the hydrogen bonds are not restricted to those between the side groups. So here this is the example. Here you can see, okay, hydrogen bonds are found at the different places. That is not only between the side groups. Hydrogen bond is between the atoms of the two peptide bonds. These are found. Hydrogen bond between the atoms of a peptide bond and an amino acid side chain. So it can be found between the peptide bond and an amino acid side chain, and hydrogen bond can also be found between the two amino acid side chains. So these are the different types of the hydrogen bond that play an important role in the structure, in the folded structure of the protein. A 3D folding of protein is governed solely by the sequence of amino acid. Depends, means what is the nature of the amino acid. In the table we have seen there are 4 groups of the amino acid: acidic amino acid, basic amino acid, hydrophilic amino acid, hydrophobic amino acid, means polar amino acid, non-polar amino acid. Okay. So these all are responsible for the different types of structure. You can see purified protein isolated from cells, exposed to a high concentration of urea. Okay, then it become denatured protein. Okay, that if we remove the urea, then it goes again, it gets its original conformation protein, so it get reformed. So that is why how we can predict the folding process, how we can analyze the folding process. Okay, so this was the structure of the protein, this was the general structure of the protein. Okay, so today we have discussed about the nature of the protein, what are the types of the amino acids. Okay, these are the types of the amino acids, and the peptide bond formation in the proteins, and these are the examples of the different proteins, and there are the different types, presence of hydrogen bonds and different types of bonds in the protein structure. Okay, so hydrogen bond play an important role in making the secondary structures also. Then again the protein get folded by the different types of bonds. Okay, hydrophobic bonds, and this is responsible for making the tertiary structure of protein, which is actually called domain. This structure you can call it domain. This is the stable structure, well defined subunit of protein, which has the biological activity. Here you can see this is the secondary structure of protein, or also you can call this the tertiary structure. This is a folded structure, but this is actually showing the secondary structure. This is formation of tertiary structure. Okay, because polypeptide chain get folded in the space with a minimum. So this conformation is obtained on the minimum energy. is a spontaneous change. It needs minimum energy. The change, any change should have the minimum energy, means minus delta G. Then only it is possible. So today, dear students, today we have learned about the introduction of the protein structures and what are the types of the different amino acids. In the next lecture, we will be discussing about the structures of the protein, detailed structure of the proteins, domains, and how these are important in the target identification, target validation, and how these are important for the drug binding and binding of the inhibitors. Now the lecture is over. Thank you.